thing and go through the slides and everything like that. Basically, this is what we're covering. Big topic. There's really good animation today. So we're really looking at specifically what's called the neuromuscular junction. That's this. And so this would be a motor nerve, any motor nerve, go pick a muscle. And effectively, and there's a bunch of them. Effectively, this is just one unit. There's tons of branches, as you will see. And this is one part of the motor nerve going to one part of the muscle, and a particular muscle fiber. That's the individual cell. Okay, so basically, what we call motor units, I guess. The way this text, most people phrase it. Effectively, the nerve signal comes down this way. So these are the events at the neuromuscular junction. We always use NMJ. It's a pretty one that's used pretty much universally. And what happens is, so when you decide I want to move a muscle, it generates what we call an action potential. That's an electrical wave that goes down the motor nerve. And before it gets to the end of the nerve and releases the acetylcholine, which is representing these little blue things over there, it basically activates a voltage-gated channel that allows calcium. This is interesting because calcium from the outside of the nerve to enter the nerve. What that calcium does, and again, it's important that this is the idea of a sequence. So the action potential goes down the nerve, opens calcium, what are called voltage-gated channels, and the calcium doesn't have any direct impact per se on the muscle itself. What it does, it enables these guys, synaptic vesicles. A synaptic vesicle. A vesicle is anything that contains something, a lysosome, a peroxisome. They're all vesicles, a membrane-bound thing. What we do with it, whether we spit it out, whether it just contains stuff for long term, is really how we define it. Synaptic means it's released into this space, which is called a synapse. When nerve meets nerve or nerve meets muscles, they're effectively what we call a synapse. Some people would say synapse. Okay. This particular one is unique because it's a nerve and muscle, a motor nerve and a muscle coming together. It's called the neuromuscular junction. So it's unique in that regard. These synaptic vesicles all contain acetylcholine. It's already made. And the acetylcholine is then released. Basically, the action of the sodium enables these little synapses to kind of come to the end of that tip of the nerve and get released. These little blue dots are that. They get released into the area. It's the tiniest space you can imagine. That's what synapses are. I mean, joint space is bigger than that. Really, this is a microscopic, not a macroscopic space. So imagine two objects that are just about touching each other, but on a microscopic basis. And what they do, once they release the acetylcholine, the little blue dots, and there's such a tiny space, they inevitably contact all of these folds that you will see very soon, basically trigger what are called ligand-gated channels or chemical-gated channels. What they do is they induce those channels on the neuromuscular junction and on what's called the motor end plate to open. And that allows, are you ready? This is a consistent relationship of sodium and potassium. You might remember it from kind of going in reverse, and basically those sodium potassium pumps resetting. This is one of the ways, and they are very, very active here in motor, more so than that. Nerves and motors are really those pumps do most of it. 3 sodiums go in and 2 potassiums go And the reverse of this, what happens by those pumps to reset it, they pump back out the 3 sodiums, they bring back in the 2 potassiums. So the net effect of this is to make the interior along the membrane more positive, or if you're one of those half empty guys, less negative. Same. So that we call those end plate potentials. Well, my handwriting's atrocious, but you're going to see this when we actually look at the PowerPoint. And if they're sufficient, they will lead to an action potential, and then the darn thing will fire. The individual cell, muscle fiber, will fire. It's a tiny piece of the muscle, and obviously we get enough of it, it generates motion. Down the road, what you will see, to remove the acetylcholine, the ACH, to sort of end all those events and effectively allow the muscle the opportunity to kind of reset and relax. Okay? Part of that involves removing the acetylcholine. As long as the acetylcholine is there, that muscle will continue to fire. We'll see that's very interesting phenomenon as we begin to look at things that can go wrong. You're going to see there's 3 ways that these little blue dots representing the acetylcholine, they can just diffuse out. It's not like it's sealed. They can diffuse out of the area. They can be broken down or metabolized. We'll see that. Or you can see an interesting process that's really important going forward called re-up. You're going to see that we break it apart, but the choline sucks back in, so we can reuse it. Very efficient. You recycle. Energy efficient. I have no idea if we are, but it's a neat analogy. And then you're going to become acquainted with the chemical that kind of makes that breakdown possible. It's called acetylcholine esterase. So ignore my bad handwriting. That's sort of the construct for today's presentation. So we call this the events of the neuromuscular junction. For those who are listening to it, I wrote it on the board. I'll attempt to take a picture or 2 and send that along with this presentation. You don't have to thank me. It's all part of the service. Sorry. I got a lot of sleep. That was the world's worst football game. Why I don't learn to go with the home team on those, like what happened to the Steelers. It's a slam dunk bet. To have my survival pool done and dusted on Thursday and be happy. So I'm going to now turn on the camera, and so we'll all be able to see it, and I will go to the slides. Easy for me to do. And so I'll reiterate that, and then you'll be able to see it. And if you just looked at this, or you're going to look at it again, we take some pictures. And please, for those in the class, take pictures. If somebody missed share them, whatever. Don't be selfish. It's a bad thing. Remember kindergarten skills, sharing, important. I have a grandson. He gets kicked out. He's like me. It's terrible. I thought I was the only kid that got sent to the principal's office in kindergarten. We're not even biologically related, but he takes after me. So, anatomy of motor nerves and the neuromuscular junction is the topic for today. So these are, the name is somatic, which means of the body, all the muscles that we basically talk about learned in a lab. The trigger, the impulse of the nerve comes from a long thread-like structure, and thread's really a good way to describe it, called an axon. We'll get into nervous anatomy, but the short course, again, you probably had this anyway, and I always draw them kind of like this. Basically, the nucleus, we don't even use that term because it's a nerve. We have to call it the cell body. There's other names for it, which we'll cover when we get to the neuro, because they use the word nucleus elsewhere. It's like what you learned about basal nuclei probably in lab, right parts of the brain. Yes. See, when we have clusters of cell bodies, we call them nuclei. When they're in the central nervous system, we call them ganglia, the peripheral nervous system. And this is the receiving end. Not all nerves are like, but the most common one is called the dendrite. It's got lots of little tentacle-like projections, like tree branches. And for that matter, so does this. The axon as it gets to the tip. The axon is the long sort of transmitter. This is the receiver, and this is the transmitter. So we're looking not just at this tip, but at branches and branches and branches. We're looking at one of those tips, these terminals of the axon. Okay. So each axon divides in branches, as I'm reading it to you, and basically each of those branches contacts a particular muscle fiber. They're spread out randomly on the muscle. That way, when we're firing a muscle, we don't always fire the same fiber because that prevents fatigue. And even when we have to lift more and more, something heavier, as we begin to lift it up, we recruit or we bring more of them into play, but we don't make them all in the same place. Again, it's all about fatigue. It's like your body does that. If you're standing in a long line to get into a movie or get into a game or a show or an event, you know how you're sitting there and you're waiting for the doors to open like that, and you're kind of shifting. Those are your muscles basically moving around to be able to do that. So that's what happens in real life. You just can't stay in one place, and the muscles just can't because they'll fatigue too much. We stand in line at church sales, rummage sales, love that. Hidden treasures. One time at a thrift store, are you ready? We found a DVD for $1 that sold for $200. You gotta love it. So each muscle fiber has one neuromuscular junction. My wardrobe is all basically, I mean, it's haute couture. I just put a little GW on it, Goodwill. Like Abercrombie and Fitch, GW, Goodwill. Moving on. So this is what it looks like. They branch out. Here's the axon. Here it's spreading out. And you notice it does it somewhat, we call it eccentrically or eccentrically, not all in a particularly discernible order. These are the individual fibers. Fine, good. Seeing this movie. So the area I call the synaptic cleft is perfectly acceptable. It just indicates the opening. I just use the generic term for it. Okay, and it's a space, and it's got a gel-like material in there. That's fine, not a big deal. But membrane-bound synaptic vesicles, and when we're talking about skeletal muscle, there's only one neurotransmitter need apply. That is acetylcholine. You'll see down the road, and it'll be confusing to talk about it now, the nerve from your brain to the spinal cord actually can give out more than one, either telling the nerve to fire or don't fire. We'll get into that down the end. It's really kind of interesting because it explains, it's one of the things that explains to you how tetanus works, why people's muscles go into spasm with tetanus. And so, I mean, this is where the concepts start to gel together, so to speak. So those little bits of surface area that I tried to dry poorly, you're going to see are called junctional folds, and they have a gazillion. As you can see here, acetylcholine receptors. So it's such a tiny space, and such a large amount just floods into the space. I used to do the demo with pixie sticks. Okay, and again, I have a pixie stick addiction. Which my daughter suggested this when I was doing a teaching demo, because you have to do a teaching and have students come in when you apply for a teaching job, so you know what you're doing. And I would just, so every time the nerve would fire, I'd cut off the tip of a pixie stick, and you have different colors. You have orange, you have blue, all that good stuff. See? A graphic display. I'm too lazy to do that now. So the axon terminal, synaptic cleft, and junctional folds are all part of this thing. And here it is. This is what the drawing is better. The cleft is microscopically tiny. There's a ton of mitochondria because it's a very energy dependent. there are a ton of synaptic vesicles. The nerve actually has a roadway when we get to the microscopic anatomy of nerves that brings these vesicles and kind of sits them there. So they're packed up and ready to go. So we don't have to worry about it. That's why the process can be relatively speaking, very, very rapid. And so it releases into this space, and you can see they're showing you these integral membrane proteins, which we call ligand or chemically gated channel, all along these folds, not nearly reflecting how many. And that's kind of where the story is. So the big picture, 4 steps. We're looking at step 1, neuromuscular junction, and then step 2, effectively how it leads to the fiber being excited. That's the animation you'll see today. You saw the second one, and we're going to go over it again. The idea of excitation, contraction, coupling, and it'll helpfully reinforce what I was trying to explain, particularly as we look more closely into it. And then lastly, probably getting to that, maybe one of those or both of these last two on cross-bridge cycling. So there's a lot there. And so that's why, and again, this is not a great slide, and that's why I put it up on the board. I can barely read it. I'm not that good. So here it says the motor neuron fires. The motor neuron axon terminal releases acetylcholine. Sounds like what I wrote. Binds to the receptors on the junctional fold of the sarcolemma. I pointed that with sarcolemma. The binding causes local depolarization. The difference between, and they're called end plate potentials. Your body is always releasing your muscles are always getting signals. Okay. It's something we call muscle tone. Even if you are asleep, there's muscle tone. Even if you are operating on somebody, there is muscle tone. When someone is comatose, that's different. Okay, then all of a sudden things are not working, and you can lift up their arm and it flops back down. Not the same when you're asleep. Their fingers, you can lift up their fingers, flop back down. That doesn't happen when you're sleeping. There's always residual muscle tone. Even when under anesthesia, when we have to lengthen a tendon or shorten a tendon, we have to account for it. Let's say there's been damage to a tendon, and we have to sew it back together. If we sew it too tightly, all of a sudden we get this. And people wake up and are not happy. And you have to go back in there and fix it. If you make it too loose, then it's floppy. So you have to know what it is as a surgeon and account for it. So the point I'm trying to make is not to demonstrate the complexity of surgery, but to understand muscle tone is something that always has to be accounted for. So these are always being released, and what that does is it keeps it ready. And so it goes back to the idea of use it or lose it. And so you're constantly using it because it keeps the system working properly. Now, all that aside, those are effectively end plate potentials. Subtle changes, and because they are limited, this electrical signal that it allows, this alteration in the membrane, very, very short-lived. If you don't have enough of it in an area or over a period of time, it dissipates because the stuff's broken down right away, as you will see, immediately. As soon as it's released, it's broken down. And so if enough of it's released in a sufficient period of time, it sort of sums itself up. And if it reaches far enough away from this juncture point, it's going to contact structures that are going to, and the fancy word is propagate, basically make the muscle fire. It'll alarm you to know that those are also voltage-gated sodium and potassium channels, but that just confuses you for now. You see what I mean? Why this is, it's really difficult stuff. So for the X number who are here, and the ones who have contacted me at least courteously saying that they were ill or couldn't attend for whatever reason, sports and stuff like that, all of which I understand, thank you. For the rest of you, where the F are you? See, I'm a dad, okay? And so my son, I had dinner with my son last night around here. It's kind of equidistant. And he just got a wonderful new job. And I said to him as a dad, would congratulate him. I said, don't F this up. I thought I'd run into some of you people, but I didn't see you up there. You know where we were at Purgatory over there in Robinson? Okay, we're in that. So it's kind of equidistant from both of us. So that's the end plate potentials. The local depolarization, and what that is, when it shifts the current inside from positive outside, negative inside, and flips it, okay, it changes the polarity. And so it flips it from positive outside, negative inside around. We call that depolarization. Okay. And so it triggers that along the area around the neuromuscular junction. You're going to see this on the animation. The action potential, if it does work, if it gets there, then it's going to trigger this thing we call excitation contraction coupling. And we will get to that. That's going to take it along. And basically what's going to happen, we're going to see is all those events that we looked a little bit at before with the T tubules and the calcium flooding in and shortening and bing, 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 and then cross-bridging. So here they are. Action potential arrives, channels open, calcium enters. That leads to the release of the neurotransmitter. If you go back in the previous chapter, and there's nothing on the test you're taking on Wednesday on V snares or T snares, but there are questions about endocytosis and exocytosis. So exocytosis is how it spits out. Remember, I told you that's where Botox works. It works particularly here at the neuromuscular junction. It blocks, though it doesn't allow those snares to latch onto the synaptic vesicles, and the acetylcholine can't spit out. And it basically paralyzes that area of the nerve, using the right term, for about 6 months. And that's why effectively people cosmetically, particularly cosmetically rather than therapeutically, or every 6 months. Now, once you start with the Botox, either you got to make a decision whether you're going to continue with it. And for heaven's sake, go with somebody who knows what the heck they're doing, if you want to go in that direction. If you did not know, it must be, if not a physician, it must be at least a registered nurse who administers that. It can't be just somebody who is and my daughter is a trained esthetician. They taught her how to do that. She's not legally entitled. I think I told you she ran one of these aesthetic places that sells, monthly packages to wealthy individuals, mostly ladies, who want to look great, and they're 50, 60 years old. So acetylcholine across the acetylcholine receptors, sodium chemically gated channels. Actually, to me, they're sodium and potassium. Opens the gate, sodium goes in, potassium goes out, and then this chemical, it's all one word, acetylcholine esterase. Esterase, ASE again is the suffix for enzymes. So this enzyme particularly breaks it apart. Acetylcholine technically in chemistry is called an ester, E-S-T-E-R. And some of the students in here, maybe it's not you folks, maybe it is, are learning a bit about that. That's really organic chemistry. So a lot of students in health sciences take chemistry as well. And here it shows you, and again, I don't necessarily think the graphics are perfect, but they're better than almost any I've seen in another textbook. And they'll go over these steps. It's a little busy, so I'm not even going to strain my eyes to try to read it when it gets all the way down here. But here you can see they're reflecting the sodium and potassium. And so it's easier to and then we're going to eventually get down here where it talks about the degradation or breakdown. They use the term degradation. I use the terms breakdown or metabolism. Doesn't matter. Every author has a different one. It's there. So if your eyes are good, or you get a magnifying glass, I expect to be able to read that. This is easier. Let's get the sound back up to where we can hear it, and I'll adjust it appropriately if it overwhelms us.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
That's the reuptake. Are you done? Sounds like it. Okay. Review. And so, I mean, that's why, I'm in love with these animations, and the best I've seen over the years. I've seen a lot of them. And so, I mean, and this is really depicted anatomically. When you actually look at micrographs of these junctions, it looks like they're almost like these like little foot pads. And so the motor end plate is what it's sitting on. So it really is, it's fairly accurately drawn. And then the whole idea, that impulse is triggering those calcium voltage-gated channels, and it allows and that's the distinction between calcium outside the cell and calcium inside the cell. For you, you just have to know which one is, that the calcium for the muscle to contract is intracellular, but to fire these voltage-gated channels, it comes from the outside. Because you're going to hear that again when we talk about the heart. You're going to hear that again. And we don't do too much on heart. We do it mostly a smooth muscle. I mean, I was looking at the exam and starting to tweak it for whenever we get this. It's going to be sometime in November before you guys take off for Thanksgiving and the exam 3. I would anticipate. You can all tell your families what an unpleasant person you have teaching you. He's really the turkey. And so here they talk about it in steps. But effectively, once the chemical is released with the aid of those calcium voltage-gated channels and its exocytosis spit out, it contacts, effectively these are ligand or chemically gated channels. I typically use the term ligand. Okay, cation just means because it's sodium and potassium. And you saw what goes on. You have, and it basically is this very consistent 3-to-2 relationship. In this case, 3 sodium in, 2 potassium out. Net effect, less negative rather, more positive. And then that creates what they call an end plate potential. If it is sufficient, it will create the action potential. That's the distinction. You're going to see a slightly different terminology when we get to the nervous system, and it's a little more relevant in the nervous system than this. And again, the terminology is very confusing for students, particularly at this level when you're just getting acquainted. I mean, I dare say most students haven't had this degree of detail in like AP biology in high school? I would not think so. That's just me. We didn't even know this existed when I was in high school. I know. And so as we get farther along, here's what happens. I mean, it's not perfectly graphic here, but they're describing the way it happens. So the three ways are diffusion, which perhaps is the least important. In this system, the most important is whatever they call it, degradation, breakdown, metabolism. Doesn't matter to me. I mean, you'll know what it is, and if you're not sure, you can ask me the question. But it breaks it down into those components. So that enzyme, acetylcholinesterase, is extraordinarily important. Okay. It's really one of the targets we use for, and I'll say this very appropriately, for both good and bad, and we'll get to that. You see, so here's the interesting thing about biology and chemistry and physics is we learn a lot from them. People of ill intent also learn a lot from them and use them for bad things. We'll get there. This is saying it's somewhat sobering when you learn about, when you're understanding the body, the people who do bad things to people, okay, they learn about it too. So the acetic acid and choline are there, they're separate. They didn't mention that there were three, they only mentioned two, but they commented the choline is reabsorbed. We call that reuptake. That is so important when we get to the other neurotransmitters, because that's the source of where most medications that treat psychiatric, psychological, nervous system disorders attack. Get to that. So that's a little bit of everything that you saw there. So we'll go through this and move on. So this is an example. A lot of things attack it. I already told you about Botox and botulism. If you did, botulism is very rare, comparatively speaking. It usually, when we see it, it's for things that are packed and canned and jars that aren't done properly. I go over this again in micro. So you've got to be really watchful of that. If you do home canning and things like that, you got to know it's got to be a sterile system. Okay, and it's really when you're not, it's using things that are low acidity. When you're putting up the parlance of canning tomatoes, you don't have to worry so much because tomatoes have so much acidity, it sort of stops the process. But a lot of other stuff can get you in trouble. The worst case was commercially was in. If you're familiar, do you know what vichyssoise is? Basically, it's French for cold potato soup. Which, if I'm sure, if you go to Paris and go to a 3-star Michelin restaurant, you're going to get wonderful vichyssoise. It's made with potatoes and leeks, lovely stock, just so nice. Dance a little parsley on top of it, but we digress. In Newark, New Jersey, hardly where I grew up in my early days, right by the old Newark airport, there was a company that made it. It was called Bon Vivant Vichyssoise. And something happened in the canning, and about a dozen people died from botulism. Because that's when you talk about food poisoning being fatal, it really isn't. The only food poisoning that potentially could be fatal is immediately fatal, better way to express it. Because this, if you don't get to it in a couple of hours, and a person all of a sudden, I can't open my eyes because those muscles twitch so fast. And then all of a sudden you have trouble breathing, and then you're in big trouble. So if you don't get it to an emergency room where someone can recognize it and give you the antitoxin, you're not surviving. And that happened. I think it happened before I was in college or around that time. I'm guessing maybe around 1970, that type of area. Maybe when I was in high school or just in college when that happened. Because I passed the plant many times driving back from the airport. Just saying. Now, that's the botulism. This is myasthenia gravis. This is a disease. It's an autoimmune disease. A little bit like we were talking about with rheumatoid arthritis. This one, again, muscles that move the fastest are the first ones affected. And so, and then you swallow, you talk, and then it becomes generalized because the person's receptors are attacked by their own antibodies. So what we use agents to prolong the activity of the acetylcholine. We block, by design, acetylcholinesterase. So we inhibit it. When we block it, we call them acetylcholinesterase inhibitors. We block that so it prolongs the availability so the muscles effectively can fire more more strongly and more persistently. Okay? Be careful when you use acronyms. So we call these ACE inhibitors. There's a really good one that really worked for my mom for a year because it works on the acetylcholine in the brain. It was one of the early treatments for Alzheimer's. And for a year she got better, and then the disease just progresses. That's called an ACE inhibitor also. And we're not even done yet. You're going to learn, if you're in pharmacology, you're in nursing, you're going to hear about ACE inhibitors also. Okay, because they work on a different acronym, ACE. Just be careful with acronyms. Be careful. So the last part of that we'll get to in a second. So the resting sarcolemma, the membrane is polarized, meaning the voltage is there. Inside the cell is negative compared to outside. We talked about that. So we start with an end plate potential. If that end plate potential reaches action potential, it'll cause the rapid shift. And you're going to see the electrical graphing called depolarization. It changes. When we go back to what the set point was, the resting point is called repolarization. And you're going to see the classic spike that's associated with action potentials. Look at an electrocardiogram, and most people have seen them either in their studies or they have looked at them on TV when they're watching a doctor show. You know, that bip, bip, and all of a sudden they go flatline. Basically, that's an accumulation of action potentials. But we talk about that, or you will, in AP2, I hope. So the acetylcholine release binds the acetylcholine receptors, albeit limitedly, and these are, again, chemically gated or ligand-gated ion channels. Sodium in, potassium out, as much. Okay, becomes more positive, local potential, end plate potential. And they're just showing you an end plate potential. And see, it's limited. And see, the big deal, does it get here? Those are, forgive me, voltage-gated sodium and potassium channels. See, that's why you're going to hate me. So if the end plate potential causes enough change, in other words, if the signal gets far enough away, we call that threshold. And you're going to see scientifically what threshold is. It will contact voltage-gated sodium channels in the membrane will open. And when they open, watch out. There's massive numbers of them. Huge amounts of sodium come in. And right after that, huge amounts of potassium go out. So it's a rapid, goes more positive and negative when you think of it in a graph. So this large influx triggers an AP, that is how RMU of it. It's unstoppable. No period, however, I don't know why, or exclamation point. And will lead to, forgive me if you're listening, RMU. Action potential spreads across sarcolemma from one voltage-gated channel to the next and the next, blah, blah, blah. And so basically this wave goes in every direction. Once it reaches there, there's so much sodium comes in, it changes it all over that sarcolemma, all over that plasma membrane, and it contacts other sodium channels. And right after that, it contacts potassium channels, and potassium flows out to reverse it. So here they are. For the time being, we're going to ignore that little gate that's there because it isn't a big player till we really have to know out of it. But down the road, you're going to learn that's called an inactivation gate. It contributes to something called an absolute refractory period, meaning you can't trigger the muscle again. It is such a small interval of time that to me, the focus point of that is much more about studying the heart. So I'm going to save it because it's really a concept that requires more advanced understanding than where the stage you are now. If you get into the heart, it's worth talking about. So here it is, generating and propagating. So it changes here, and as soon as it changes here, it changes there, and then right away it resets this one. So it's how fast can you contract your muscles? Rapidly until they fatigue. Nerves even faster. Okay, the heart's different. So repolarization, restoration of the channels, the voltage-gated channels close, potassium channels open, the efflux rapidly brings it back to the initial resting membrane, and then there is a refractory period. We're talking fractions of a second. We're really talking in this milliseconds. So muscle fiber cannot be stimulated for a specific amount of time until that is complete. In other words, it has to get to a point where we can re-trigger it again. And then now understand, this is where you're going to hear about electrical events precede chemical events and also precede mechanical events. So we electrically reset it. Sodium flowing out, I beg your pardon, sodium rushing in and potassium flowing out super rapidly in large amounts resets it electrically. But now you got a whole bunch of extra sodium inside and a whole bunch of extra potassium outside. And you got to flip it back to its normal condition chemically. And that's why so much of our energy is because of these sodium potassium pumps. The sodium that came in is pumped out, the potassium that went out is pumped back in. A lot of energy every time we do something. And whether you're thinking about something, but mostly when your physical actions are really doing the most of us. And sometimes it induces something that we're very familiar with, fatigue. And there's reasons why some of us fatigue more than others, but that's a lot of lectures put together. So repolarization is the restoration to its initial state. You can look at these. The whole idea is if this gets past that motor end plate and contacts just a couple of these voltage-gated sodium channels, it'll just send it in every direction. It could touch the fiber here, that long cylinder, touch it here, and the whole fiber will be triggered. That is not what happens in nerves. Okay? And in nerves, we're going to start to look at that inactivation gate. That is why the impulses are directional. They go from the cell body all the way down to the tip, that transmitting end. Very, very directional. That's boop, boop, like that. So the refractory period is probably to right about here. And so what are we looking at in milliseconds, thousandths of a second, less than 10. Nerves are faster. Just give you an idea. That's the muscle fiber. And different muscles are faster than other muscles. Cardiac muscle, totally different. Once your heart beats, it's almost a quarter of a second before it can beat again. Okay. So then we get to what we covered the other day, the excitation, contraction, coupling events, blah, 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 blah. So the excitation, contraction, coupling, we transmit it, excitation, and then now we're going to do something else. We're going to change that wave of action potential and electrical activity into mechanical activity, which is contraction. So propagated means just going along the membrane, and then we get to the T tubules, which I'll talk about again, and all of those things, and the calcium release, and it's brief and ends in contraction is seen. And that's when we're going to start looking at all of these things that are there. I don't want to start these today. I mean, I got this done a little quicker than I thought, but we'll end it here, and we'll start with the excitation, contraction, coupling on Monday, because I know it's a lot to digest. Thank you very much. Get a few minutes early, we're off.